My name is Nick Zayats. I'm an associate professor of pathology here at Case Western Reserve. And also I'm the chapter president for Sigma Xi, which is here at Case Western Reserve. And if you don't know, Sigma Xi is a multidisciplinary international organization whose main interest is in science and education. I might add that the Case Western Reserve chapter is one of the oldest chapters in Sigma Xi. And we would like to encourage any of you in the audience to join our organization. If you want to, you can contact me or see me afterward. And we're happy today, the Sigma Xi chapter, some of the officers sitting in the office, just want to recognize them. Ken Street is the secretary-treasurer. Tom Mortimer is, saw him here, he's in bioengineering. And Darren Croft is also on the board there. And the other thing I want to mention is, one thing we're doing interesting, if you haven't heard, we're doing science cafes on Monday. It's down at Great Lakes Brewery, and we have a number of engaging topics that we've had for the past year. And go to the website at Case Western and type in Science Cafe, and you'll see the schedule there. Actually, what is the next program, David?
This Monday night, it's Origin of Life on Earth.
Yeah, Origin of Life on Earth. One of the colleagues, Neil Greenspan, who's here for pathology, will be talking as well. Neil is back there. So today's speaker is being sponsored by not only Sigma Xi, but the evolutionary biology program here at Case. Our speaker comes to us from the Carnegie Institute of Washington Geophysical Laboratory, and he is a Clarence Robinson Professor of Earth Science at George Mason University. Dr. Robert Hazen has an extraordinary career. He received his BSN SM in geology from Massachusetts Institute of Technology, went out to get his Ph.D. from Harvard University in earth science, and then studied in a NATO postdoctoral program at the University of Cambridge. He then joined the Carnegie Institution's research effort. He's published over 300 articles, 20 books, and by the way, if you're interested, 2 of those books are outside in the lobby there. He is also an avid follower of music. In fact, he is a trumpeter with the, I have to get it right here, with the, I'm not sure the exact organization you're with right now, okay, but has had numerous ensembles in New York City, Boston, Washington, the Jeffrey and Kirov Ballet, Boston Symphony. So you can see he's got this background that's enormous, not only in the science and the geological science. In regard to today's talk, the genesis, the scientific quest for life's origin, as we continue our series on Darwin. So without further ado, I would like to introduce Dr. Robert Hazen.
Thank you. Genesis. For some people, the first chapter of the Bible is explanation enough for the origin of life, for the evolution of life. Scientists seek for something more. They seek for a natural explanation, an explanation consistent with the laws of chemistry and physics. And I think there's no subject for which there's greater tension, greater conflict between science and religion than this search for origins. Origin of life, the origin of humans. And you can understand that. How is it possible that we are created in God's image if we're just simply the product of random chemical reactions or some random evolutionary process? How do we address this? As a scientist, I'm convinced that there's a natural explanation. And we seek for this explanation through the process known as chemical evolution. Looking for the transition from geochemical simplicity to biochemical complexity. And we do this by looking for a sequence of chemical events that can be studied in a laboratory environment. That's our challenge. That's our task. But there's another point of view. There are other points of view out there. And you all know one of them is intelligent design. The idea that life is irreducibly complex. So complex, in fact, that it's impossible to imagine a natural process from going to simplicity of chemistry to the complexity of that first cell. The argument goes like this, that if you were walking along in the woods and you saw a wristwatch lying on the ground, you'd reach down, pick it up, look at it, and you could see from its complexity, from the nature of its materials, from the intricacy of its assembly, it could not possibly have arisen by any natural process. It had to have been engineered. It had to be created by an intelligent designer. So it is, they say, with the first cell, because a cell is infinitely more complex than even the most intricate wristwatch. And so this hypothesis requires a combination of indeed some natural processes, but supernatural processes as well. Supernatural processes that cannot be understood by scientific experimentation. Now we may ask, is intelligent design scientific? And what I have to say is, in some ways, on the one hand, intelligent design does make testable predictions. This point is often overlooked. It predicts always in the negative, that there is no natural process by which the flagellum of a bacteria could arise spontaneously. There's no natural process by which an eye could evolve spontaneously. Indeed, there is certainly no natural process by which the first cell could have arisen from non-life. And you can test these hypotheses because if in a laboratory experiment we can go and we can synthesize, we can make from scratch a bacterial flagellum, or an eye, or a cell. And we've disproven a basic tenet of intelligent design. On the other hand, intelligent design fails one important litmus test of science because it depends on explanations that are supernatural, processes which are inherently not testable in a laboratory, things that we cannot make independent observations and verify them. We cannot inherently know who that intelligent designer is, the creator is. And so in this sense, intelligent design fails the test of science. Now, this leads to the debate. You've all heard about this. There are some leaders who have expressed, let the children decide. Teach the controversy. Because after all, there's intelligent design, there's evolution, these are two, weigh one, weigh the other as if they're equally balanced choices. That's one answer. The other, though, which every scientific society and many scientific leaders have argued, is no, we can't do this. In a public school science course, you cannot teach intelligent design. You must teach Darwinian evolution. And so it's easy to see how we as scientists appear closed-minded. We're the ones who are scared to address that controversy. We're the ones who are hesitant to have an open and honest debate, and it makes us look bad. What are we to do? What I do as a scientist is I go into the laboratory and I design a research program. I say, if we can show in the laboratory a series of experiments that goes from geochemical simplicity, step by step by step, to the complexity approaching a living thing, then we have invalidated the claims of intelligent design. We have shown a natural process by which life can arise, and we have therefore solved that problem. Let me give you an analogy to this. Stonehenge, the great prehistoric monument on the southwestern coastal plain of England, Salisbury Plain, built some 4,500 to 5,000 years ago. And there are those who claim it was built in part by space aliens, if you will, intelligent designers. Now, there's absolutely no way that any anthropologist anywhere can disprove that claim. It is possible that space aliens came perhaps with flying saucers and they helped cut out the stones, and these massive stones, which it's difficult to see how any primitive culture could have lifted them up. They lifted them up and set them down, and so Stonehenge was built this way. However, it is also possible to imagine a way that a primitive society, however difficult, however much time it took, could have used their ingenuity, used rope, used wood, used rock, time, 1,000 people working together for collective good, and produce Stonehenge. Using the scientific criteria of Occam's razor, the least number of assumptions that you have to add to an explanation, this is a better explanation. We know about all these different processes, and we can imagine ourselves collectively going and doing this without the use of space aliens and flying saucers. However, we can't prove the opposite is true. The simpler explanation, however, is the one we should accept. And so again, I tell you, if I can show you a process by which we go from chemical simplicity to complexity, then why resort to intelligent design? So that's the challenge we have as scientists. That's our best rhetorical argument. That's our best weapon against this kind of thinking. So how do you go from simplicity to complexity? And the process that we see around us every day, all the time, in 1,000 different ways is emergent complexity. It's a process by which numerous individual particles, acting only in response to the local conditions of that particle, collectively result in new patterns, new structures, new phenomena. And we see it all the time. Sand grains. Individual sand grains responding only to the force of gravity, the collective force of the other particles in contact with it, and the lateral force of water or wind collectively form patterns of ripples and dunes. We see it in much larger scales in the spiral arms of galaxies, each star responding only to its local gravitational environment, and yet collectively these magnificent spiral structures, the larger scale structures. No one star is a spiral arm. These are large-scale collective properties. And of course, the collective property of your billions of neurons complexly interconnected, creating consciousness, allowing us to have this conversation today. A great example, one of my favorite, is the collective emergent property of the social amoeba, Dictyostelium, slime mold. Slime mold lives most of its life as an isolated single cell, a eukaryotic cell. There's the nice nucleus of one example. And under certain circumstances, when the conditions in the forest floor, if you become starved for nutrients, or the temperature, the humidity changes in certain ways, chemical signals are sent out. And each cell responds to those signals going towards the gradient. And so you begin a collective migration. And then these collected organisms, the single cells, literally crawl across the forest floor. That's about the funniest thing you're going to see today, so you can laugh now if you want. Okay, thank you. And then what they do is they form these stalks, and a whole new generation of single-celled spores are released, and a new generation of Dictyostelium comes about. Every single cell is only responding to its local environment. It doesn't know that it's part of this larger structure, and yet the collective behavior is the structures, the properties, are emergent. That's what we mean by emergent phenomena. And again, just to underscore the point, here's a single neuron in your brain. You have billions of these interconnected in a network with electrical impulses of varying strength. They form that neural network. The collective result is consciousness. And of course, no single neuron is conscious, but the collective emergent property is quite different. Now, what I'm going to argue is that the origin of life can be viewed as a sequence of emergent phenomena very much the same, each one where collective properties of individual particles come together and lead to an increasing layer of complexity. We see this at every scale. Here's the consciousness idea. But in terms of space, we see it in the spiral of the ring structures of planets, the hourglass structure of exploding stars, the spiral arms of galaxies, this kind of simple patterning. We see it in life at the scale of molecules. We'll come to these lipid bilayers a little bit later. And of course, there's the cellular structures and the collective social behavior of colonial animals like ants. Here's a group of ants tending aphids. Isn't that amazing? Each individual ant does not say, oh, I'm going to tend aphids today. It's just responding to local chemical signals, and yet the collective behaviors of ants are quite remarkable in this way. And of course, social phenomena, the kinds of collective things that we as humans do. So there's a marathon, and one of my very favorite, of course, is the symphony orchestra, a collective phenomenon where each individual can play individual notes. The collective phenomena of the symphony is vastly greater. Okay, so let's think about origin of life in this context of emergence. And I want to lay some ground rules first. We're not going to use exotic chemistry. We're going to use simple organic chemistry, the chemistry of carbon. Life is carbon-based, and there's no reason not to begin with simple carbon-based molecule. And the building blocks, the raw materials. You have the oceans, you have atmosphere, you have rocks and minerals. So let's not resort to any exotic ingredients, just the basic stuff you'd have on any planet like Earth. And then the origin of life, we're going to suggest, requires a sequence of emergent steps going from simplicity to complexity, each step involving the interaction of many small particles to make larger and more complex particles. And I can vision the origin of life. You may think of it as a continuum, but let's think of it as 4 separate steps. And the advantage here is that each of these steps can be studied as a sequence of experiments you can do in a graduate student's lifetime in a university. And that's important. If you want to get a degree in something like this, you don't want to have to spend 50 years doing it. So first we want to make some biomolecules, just basic building blocks, amino acids, sugars, lipids from raw ingredients. Second thing, we want to see those molecules selected and organized into the larger structures of life, membranes and polymers. You then need to have a self-replicating system, a set of molecules that makes copies of itself at the exclusion of all other molecules. And finally, natural selection comes into play as molecules make copies of themselves, they compete for resources, and the selection process of competition winnows out the less efficient, rewards the more efficient. That's the sequence of steps. So let's go through these one by one, and I'll show you some experiments and how we can make progress on this really exciting subject of life's origins. First step, the one we understand best, the emergence of biomolecules. And what you're starting with, the starting materials, are simple volcanic gases. Carbon monoxide, carbon dioxide, methane, ammonia, hydrogen disulfide, water. Simple molecules, some of them with a single carbon atom. And what you want to do is step by step form carbon-carbon bonds. That's really the key, making larger organic molecules from the simple building blocks. So things like acetic acid going to pyruvic acid with 3 carbon atoms and so forth. Remember, pyruvic acid, we'll come to back to that a little bit later, but this is the kind of thing we want to do. And this has been known to be actually quite easy to do for more than half a century. I think most of you will recognize the classic Miller-Urey experiment, an experiment which so beautifully mimics early Earth's environment. Water representing the ocean, a mixture of gases representing the atmosphere, little electric sparks representing lightning. You set this thing going, you gently boil that water, you get convection going around in this cycle. And as you spark this, very, very quickly, in a matter of a day or two, you begin to see the solution turning yellowish and pinkish, and then browns and black goo starts dripping off the electrodes, and all this stuff comes out. And what it is an amazing distribution of molecules that are formed very quickly that are the building blocks of life. The amino acids that make proteins, sugars of carbohydrates, the carboxylic acids, including key molecules that are part of metabolic cycles, as well as molecules that can form membranes, and basic building blocks of the nucleic acids, DNA and RNA. And the reason people were so excited and so quickly assumed that we had solved the key problem, the origin of life, is when you look at this list, it looks very much like a balanced diet. Because this is what we're made of. So that's what Miller discovered in the early 1950s, and in a whole sequence of subsequent experiments using a variety of atmospheric compositions and different geometries. In fact, there was a paper that came out by some of my good friends and colleagues just last week in Science, which revisited some of Miller's original experiments using a different set of conditions and finding even more amino acids that they found in the original. Modern analytical techniques allow you to see that this was a very, very robust and productive way of going about it. But there are other ways as well. Lou Amendola, my good friend at NASA Ames Research Laboratory in California, studies what happens in dense molecular clouds in deep space. These are places where with radio telescopes we can actually see evidence that there's all kinds of organic molecules. And when they talk about dense molecular clouds, we're talking about extremely cold environments, just a few degrees above absolute zero, and an extremely high vacuum, a much higher vacuum than you can make in any laboratory environment, and yet integrated over a very large volume of space, they look like clouds. So for example, the Eagle Nebula is an example, and this is a vast, vast structure our solar system would just be about the size of that dot in the middle of here. So we're really talking about immense structure. But if you look at the radio signals from this, you find things like sugars and formaldehyde, amino acids indeed, which are difficult to detect. So what they do at NASA Ames is they simulate that environment by having a vacuum chamber that's chilled down to 10 degrees above absolute zero. They irradiate small molecules with ultraviolet radiation, and they produce amino acids. They produce sugars. They produce building blocks of DNA and RNA. Turns out that happens in deep space. And in our laboratory, we do the exact same thing, but we look at an environment on the ocean floor. The geophysical laboratory where I work is specialized in doing high pressure, high temperature experiments. So this was a niche for us to explore that other people hadn't looked at. Now, we're not advocating this is the only answer, just one more environment where things might happen of interest. And so we mimic the high temperature, high pressure, mineral rich environments of these black smokers. What we do is we seal small amounts of compounds like carbon dioxide and water with minerals in a gold tube. We subject those gold tubes to high temperature and high pressure. A lot of this work was done by my late colleague, Hat Yoder, a fantastic mentor of mine. At the age of 75, he was still going into the laboratory every day, working 8 hours a day. He had retired, but what could be better than doing this for a life's work? He sadly got a little minor infection, went into a hospital. The microbes killed him when he was still very active. It's a very, very sad loss. But we carried on that work. And what we do is we open up those capsules and see what happens. George Cody, my colleague, organic chemist, does the analytical work. Here you see with a gas chromatograph, a GCMS, where you basically analyze the molecules that come out of these experiments. And let me share with you the kinds of things we're looking for. We want to make carbon-carbon bonds. I'm going to show you two different ways that organic chemists could think about making carbon-carbon bonds in these experiments. One is a way of just building hydrocarbons that are longer. So you start with natural gas, methane, and you just build up by adding CH2 groups, making the chain longer and longer. That's a classic process which goes by the name Fischer-Tropsch synthesis. And you can build very long molecules this way. Some of our very first experiments were looking at Fischer-Tropsch synthesis. What we did was just take a capsule, used carbon dioxide, hydrogen, and water, had a little bit of iron metal in this case, but you can use all kinds of iron minerals that does the same thing. 300 degrees centigrade, 500 atmospheres, conditions very typical of a black smoker for just 24 hours. This was one of our very first experiments 12 years ago. We opened up the capsule, and what you see here is the trace of a gas chromatogram where we basically pass these molecules through a long column. The small molecules go through the column rather quickly. Bigger molecules take longer. And so as a function of time, which is this horizontal axis, you see the small molecules coming out first, and each of these peaks is a different molecule. The N stands for the number of carbon atoms that have been built up into a chain. And amazingly, overnight, we produce molecules 17, 18, 19, 30, and more carbon atoms in a long chain. Furthermore, all these little bumps and wiggles here, some of these are branching molecules, some of them are alcohols. We're making big molecules from small building blocks by Fischer-Tropsch processes in hydrothermal conditions. We know this occurs in Earth's shallow crust. We know it occurs in mid-ocean ridges. This is a basic process for making organic molecules from CO2. Now, there's another way of building large molecules that uses a slightly different chemistry. This is called hydroformylation. And what it is you're basically inserting a CO group, not a CH2 group. So here we go from 2 carbons to 3 carbons and so forth. And this actually can lead you into the kinds of molecules that are used in metabolism. So for example, pyruvic acid, carboxylic acids, these will be familiar names to you if you've done biochemistry. So this is a different kind of chemistry, but it turns out that this also is promoted by certain minerals in hydrothermal vents. And here we have what looks like a rather messy diagram, but let me see if I can walk you through this. In blue, Fischer-Tropsch synthesis. That's making longer and longer molecules by inserting CH2 groups. In red, hydroformylation, making longer and longer molecules by inserting CO groups. And what you see is that different minerals, these are all minerals which occur in hydrothermal vents, lead and cadmium, zinc, copper, nickel, cobalt, iron, sulfides. Oxides do the same things, by the way. And you'll see that some minerals are very good at Fischer-Tropsch. Some are very good at hydroformylation. Some do both. And indeed, you can understand this, the chemistry of this having to do with the configuration of the outer electron shells, that some link onto CO groups more efficiently, some link onto methyl groups more efficiently. But the fact is, what we're seeing here is in a submarine vent, you have all these different minerals, you have fluids circulating over them, you have reactions going on constantly. You add a CH2 group here, you add a CO there. If there's nitrogen present, you can add amine groups. You can have sulfur added into it. You can make this diversity, this incredible complexity of organic molecules in hydrothermal vents. And depending on the sequence with which the molecules see different mineral surfaces, they keep adding one group or another, branching chains, rings. You can form a myriad of molecules. All the kinds of biological molecules you might want can form in these hydrothermal systems. They're forming today on Earth. They certainly formed in the ancient past before life was here. Conclusions, step one. This is the easy part. This is what we know for sure. There's tons of different ways of making biological molecules. And as a mineralogist, I am pleased to announce that minerals certainly played a role in this whole process. Making the molecules is easy. Making amino acids, making sugars, making lipids, making bases. We can do that. Problem solved. Step two is a little trickier, because you can't make life just from a bunch of random molecules. Life is incredibly selective. It is incredibly idiosyncratic. There are in Beilstein, a list of organic molecules, some 6 million small organic molecules known. A recent list of all the small molecules in E. coli numbered only about 500. So how do you go from the millions of possible molecules, or if you will, the tens of thousands of molecules we know we make in these experiments, to 500? How do we go to just those 20 amino acids? Why ribose and not the other pentose sugars? And on, all the questions we can ask. So here's the problem. Prebiotic synthesis makes all different kinds of molecules. Life is highly selective. How do you select and concentrate just the right molecules? What is the process by which that prebiotic soup, which not only was incredibly diverse in its molecules, but was hopelessly dilute, the prebiotic soup, the ocean, no matter how much organic synthesis you can imagine, it still was much too dilute. How do you do this? Well, there are a couple of possible ways, and I want to give you a sense of how our experiment showed this. One of them is called self-organization. There are some molecules that will find similar molecules and build structures automatically. Let me tell you about an experiment that we thought we would do that was going to lead to a science paper. You know, you shouldn't do this. I confess, you shouldn't do this. You shouldn't say to your colleagues, well, we're just going to do this one simple experiment and we'll write up the paper tomorrow and we'll send it off the next day, because it's so easy. And the experiment we were going to do was one that Harold Marwitz, our good colleague, said, take pyruvic acid with 3 carbon atoms, add it to CO2, and you put it under pressure, and the CO2 will bond to the pyruvic acid, and you'll make the 4-carbon molecule oxaloacetic acid. And that's a key step in what's called the reverse citric acid cycle. I'll show you that later. It's a metabolic cycle. If you can show that step, then you've shown a key step in going from simplicity to a metabolic cycle. And maybe minerals will catalyze this, and this would be a great discovery. So we put pyruvic acid and CO2 and water, very modest conditions, 200 degrees centigrade, a couple thousand atmospheres, 2 hours. We did not make oxaloacetic acid. Instead, our simple little drop of colorless fluid, when we opened up the capsules are all swollen up like they're ready to burst. And so I said, uh-oh, we better dip this in liquid nitrogen and freeze what's ever inside, because the capsule might explode if we pop it open. So we did that and froze it in liquid nitrogen, snipped it open, and nevertheless went, you got a big explosion. And the pieces of gold that clipped off the end, in some cases, went flying across the room like a projectile. And then you drop the capsule into a glass vial, and you sit there, and as the thing unfreezes, it starts going, You hear this hissing and bubbling sound, and then this yellow-brown oily goo starts foaming and pouring out of the capsule and coating like an oil slick the bottom of the capsule, this goo. And then you start smelling this thing. And it's astonishing. At lower temperatures, it smells like molasses. There's a real sweet smell to it. At about 300 degrees centigrade, though, that you get a smell that's exactly like Jack Daniels. and it pervades the lab, and people come by and they smell and say, is there a party going on in here? Can I have some? But what that means is we made some really interesting molecules here. We made some organic molecules that we didn't expect and clearly have this characteristic of being large enough to interact with the sensors in our nose. So we then do a gas chromatogram with George Cody, and we get this mess. There are thousands upon thousands of different molecular species here in this GC. Every one of these peaks is a separate compound, and this hump represents literally thousands of different things. So we got a whole mess of stuff. And we said, this is really discouraging. We hope we had done an experiment that would have direct relevance to the origin of life, because if you go from 3 carbons to 4 in a concerted, systematic, highly productive way, that could be a step that life uses. That looks like a metabolic step. If you go from 3 carbons to a mess, it has nothing to do with life. In fact, you've made much too much stuff, and you're just wasting your pyruvic acid in something that wouldn't have any relevance. So we said, how in the world is this going to help us out? And along comes my good friend Dave Deamer, University of California, Santa Cruz, who spent his life studying self-organization. He says, this goo, this yellow-brown oily goo, looks a lot like some of the stuff that I've extracted from the Murchison meteorite, and it might self-organize. And my colleague Marilyn Fogel, who also analyzes molecules like this, said, yeah, I could look at these and see what kind of size distribution you have. Do you have 20 atoms, 20 carbons, or 30 carbons, or other sizes? And she was able to show that the hump there was really concentrating around 10 to 12 carbon atoms, which is a length of a chain, which is very good for self-organization. And so they said, maybe you're going to get a situation like this. Now, this is a schematic of what the molecules, the phospholipids that make cell membranes look like. And they have an end that's attracted to water, hydrophilic. The other end is this bobby pin-like thing, is repelled by water. So if you stick a lot of these molecules in water, they form a bilayer. The hydrophobic ends get away from water. The outsides are hydrophilic. And so you can form a vesicle-like structure, a cell-looking like structure that has an inside and outside. And lo and behold, we put this yellow-brown oily goo in water, and it just spontaneously forms. By the way, these things are autofluorescent too, which is really cool. So they look just absolutely beautiful under the microscope. And they're about the size and shape of cells. Now, they're not cells, but what it means, what we're seeing here, is what I think is a universal phenomenon in the origin of life. You will spontaneously self-organize structures like this. In that whole diversity of molecules we make, some of those molecules will find others of a similar ilk. They will come together, they will organize into these spherical structures. The formation of a membrane-like structure, therefore, is not a mystery. This is something that's deterministic. We find this with the Murchison meteorite. We find this with materials synthesized by Lou Alomandola at the deep space environments. We find it in our hydrothermal experiments, in the deep ocean environments. Problem solved. But there's another problem associated with this kind of selection and organization, because an awful lot of the molecules we're interested in, amino acids, sugars, are highly soluble in water. And therefore, they're not going to do this kind. They don't have any hydrophobic parts. They're hydrophilic. They're going to dissolve in water. You're not going to be able to concentrate them in this way. So you need to find another way. And in particular, things like amino acids and sugars have an additional selection problem. A lot of the molecules in biology come in mirror-related pairs, like your right and your left hand. Life only uses left-handed amino acids, not the right. It only uses right-handed sugars, not the left. How in the world do we get that kind of selection out of a prebiotic soup that was so dilute? So here's the idea. Left-handed and right-handed molecules are mirror images of each other, a central carbon atom with 4 different linking side groups, and they can be arranged clockwise or counterclockwise. The key question that we want to ask in origin studies is, how did life become homochiral? That means all the same handedness. But this is also a huge question in industry, and so our collaborators in this work are chemical engineers, because you pay a lot of money for drugs that are chirally pure. chirality is important. And the reason is because left and right-handed molecules have different properties in terms of their interaction with you. Limonene, the right-handed version, smells and tastes like oranges, is used in that capacity for flavoring. But the left-handed version smells like lemons because our mouths have chiral reception centers and different-handed molecules are going to interact differently. A more sinister example is thalidomide. You've heard about this, and it turns out that the right-handed form of thalidomide helped cure morning sickness in women, but the left-handed form caused birth defects. And it was giving women thalidomide where it automatically racemized. That is, even if you took a pure chiral form in your body at body temperature, it would gradually flip from right to left and then cause that tragic consequence. So chiral selection is important. Now, the hypothesis that we have has to do with natural chiral environments. You have a mixture. You make in prebiotic processes I've told you about, you make equal amounts of left and right-handed molecules, but life demonstrates this selectivity. Here's the question: what's the mechanism of symmetry breaking, as it's called? And what we propose is that the symmetry breaking could have been accomplished by minerals which intrinsically have their own chiral surfaces. They have handed surfaces. The most obvious choice, and has been used for more than 70 years, is quartz. Quartz is a common mineral of beach sand, and it is the only common mineral that comes in left-handed and right-handed varieties because its structure is helical. It has a helix of silicon oxygen 4 groups, and it can either spiral up to the left or spiral up to the right, hence left and right-handed quartz. Now, in the past, many researchers have used quartz, and what they do is they take a nice crystal like that and they grind it to the finest powder they possibly can. They do that to increase the surface area. But it's a terrible thing to do. Not only does it destroy a really nice crystal, which as a mineralogist I hate to see, but you destroy all the information that you might have obtained from the faces. And here's why I say that. Here's a specimen of quartz I collected when I was a teenager in Paterson, New Jersey. If you look closely at this, this is one crystal. It's about 1 millimeter across, has 6 triangular faces, and you'll notice that 3 of those faces are coated by iron oxide, and 3 of them are not. And the reason is quartz has 3-fold symmetry, and the surface structures of these 2 faces are very different. Indeed, every surface has a different structure from every other surface. And so if you grind up the crystals, any information that's specific about how a molecule is binding to the surface is lost. The only way, indeed, you can study selection, chiral selection, for example, on a mineral surface is to study individual crystal faces. And so we have been systematically looking at common rock-forming minerals that have chiral faces. Here's feldspar, the commonest mineral in Earth's crust. It has right and left-handed faces, mirror symmetry structures. Here's diopside, the commonest iron-magnesium mineral in Earth's crust. It again has right and left-handed faces. And my favorite, calcite. Calcite is the mineral of clamshells and snails. It forms coral reefs. And here you see a mineral that has every face is either left-handed or right-handed in this common crystal form, this common pointed crystal form. And furthermore, these faces have wonderful topologies of atoms, perfect for sort of selecting and concentrating, pulling out of solution molecules of different sizes and shapes. So our hypothesis was if you take a crystal like this, you expose it to left and right-handed amino acids, some of those amino acids are going to concentrate on the left-hand face, the other handedness will concentrate on the right-handed face. An experiment would take a fist-sized crystal, you dump it into a beaker with a 50-50 mixture of left-handed and right-handed aspartic acid, that's one of the amino acids, let it sit there overnight, pull it out, face by face, put a very small amount of hydrochloric acid which strips off the top layer of mineral, pulls off the amino acids, put that with a pipette into a little glass vial, and then analyze the left and right-handed ratio of amino acids. And sure enough, what we found is that left-handed amino acids concentrate on left-handed faces, right-handed amino acids concentrate on right-handed faces. This is a way of selecting and concentrating in local environments amino acids of one-handedness or the other. Now, to be sure, there are equal numbers of left and right-handed faces, so whether you ultimately end up with right-handed life or left-handed life, I think is just a matter of chance. You have to have the origin of life at some place, some time, some microenvironment. That's the thing that began to make copies of itself and self-replicate and take over and become more efficient. In our life, it happened to be left-handed amino acids was the best at this. That was just a chance thing, I think. But whether that's true or not, you can see that minerals provide one solution for this problem. And so my conclusion in step 2, we don't know all the details for sure, but there are 2 ways that we know are important in the selection and concentration, the organization of these molecules. Self-organization, as we see the lipids, mineral surfaces, and concentrating, selecting out of solution in the case of mineral surfaces and amino acids or sugars. Step 2. Okay, here comes the difficult one. This is the big one. This is the holy grail, if you will, of origins research, making a self-replicating molecular system. There are some of our colleagues who feel strongly that looking at life today, looking at the core metabolic cycle, the replicating cycle in all life today is the answer. The TCA cycle, Krebs cycle, citric acid cycle, depending on how you want to call it, can be run in reverse of the way that you've probably normally learned about it. And it's an engine of synthesis. You start with CO2 or pyruvic acid or acetic acid, and you just add small molecules. You go around all the way to citric acid. You split this into 2 plus 4. And then you have 2 cycles, then you have 4, then you have 8. Each time you go around the cycle, adding only small molecules, you build this up. If we could get this cycle to run in a test tube, perhaps using mineral catalysts, I think everyone would agree we have the origin of life problem pretty much solved. This would be the first metabolic cycle. In fact, there are 11 steps here. We have 9 of them working using just common mineral catalysts, minerals that occur on those hydrothermal vents, for example. There are 2 in particular. This one I mentioned to you before of adding CO2 to pyruvic acid to go to oxaloacetic acid. 1 plus 3 goes to 4. We can't get that to go. And I don't know what the answer is going to be. Maybe there's some other path. Maybe we're got the completely wrong cycle and there's some other cycle. But this is one way of thinking about it, that there's some simple metabolic cycle that if you can get it to go, it'll start making copies of itself. It'll be autocatalytic and just take off. And then all the other synthesis, all the other biosynthesis, as you probably know if you've taken that biochemical course, comes as side groups. Amino acids and lipids and sugars all just come off through other reactions off this. Here's my good colleague Harold Marwitz, who feels this is the deterministic aspect of life's origin. He argues this has to be on any planet, any moon, anywhere in the solar system, anywhere in the universe, indeed, that life begins. This is the cycle, and here's that reductive citric acid cycle and some of these side reactions that make the other molecules of life. Very powerful core biochemistry, perhaps pointing to this origins. That's not the only idea out there, though. There's another idea Stewart Kaufman has proposed. When he was at the Santa Fe Institute, he claimed that you could have an autocatalytic network of molecules, sort of a spaghetti diagram like this. And this is rather simple. He said that diagram maybe should have 1,000, maybe 10,000 different molecules. The idea being that each molecule in some way promotes or catalyzes the reaction of other molecules in the network. And so you have a network in which every molecule is somehow supporting the formation of other members of the club, and you exclude everything else. And as a result, the members of that club, of that network, increase in concentration. It sort of becomes a self-replicating morass of molecules, and everything else is shut aside. And then subnetworks start competing with each other, and you get smaller and smaller networks that become more and more efficient. That's the idea. Now, there's no chemical meat on these bones. It is just an idea, but it's another idea about self-replication that's out there. And there's a certain kind of persuasiveness if you think about an early milieu that has so many different molecules. Maybe something like this is the way things get going. We just don't know, and it's very hard to design an experiment that studies 10,000 molecules at a time. And then there's the idea that maybe the first living system wasn't a metabolic pathway, but in fact was based on RNA. And the reason RNA and the RNA world is such a powerful idea is that RNA acts both as an information-carrying molecule, genetic molecule, but also can serve as a catalyst. So it does everything that life has to do, catalyzes reactions and carries information to the next generation. And so this idea of the RNA world has been raised. The trouble is that RNA is a very difficult molecule to synthesize from scratch. There is no prebiotic way that anyone has come close to making RNA molecules. You can make amino acids, to be sure. You can make bases. You can make sugars. But to stick RNA together with the phosphate and the sugar and the base in just the right way with just the right linkages, and much less to link them together in a polymer, is extremely difficult to imagine. So what happened between that prebiotic soup with all the molecules in the RNA world? This idea of the pre-RNA world is one of the biggest mysteries in prebiotic science. I'm going to take you on a weird journey right now, because what I'm about to show you is highly speculative. It's not been demonstrated experimentally, but it somehow encapsulates or epitomizes the kind of creative thinking that goes into origins research, where someone can come up with a clever idea and put it out there, and then the community can look at it and eventually test it experimentally. Sadly, this idea has not been tested experimentally. But I want to introduce you to Nicholas Platts, an Australian scientist who came to the United States and at the late age of 40 got his Ph.D. at RPI, and he got his thesis largely approved on the basis of an idea, a clever chemical idea about what might have preceded the RNA world. Here's what Nick said. He said, if you look in the universe, there are vast numbers of compounds called polycyclic aromatic hydrocarbons, or PAHs. These are the basic molecules that make up soot from a diesel, fumes, or if you're barbecuing or grilling, you see that. And in deep space, you find vast quantities of these. This is basically an infrared image at the wavelength of PAHs. And the implication is that there's a vast quantity of these cyclic carbon molecules throughout space, and that any planet or moon is constantly being rained upon with these things, and that would have been part of the building blocks. And he said, wouldn't it be nice if all those carbon compounds were actually of use in the origin of life, as opposed to just a way of locking up carbon in a useless form? And he said what would happen is if you stuck polycyclic aromatic hydrocarbons in a water environment, they actually act like amphiphiles. That is, they have portions that are repelled by water. That's the flat part. But the edges are attracted to water. And then how do you assemble, how do you self-assemble a bunch of flat, round molecules to do this? And what he said is you just stack them up like a bunch of pancakes or a bunch of plates. This is called discotic self-organization. And you do this, and it turns out the spacing between these disks is exactly the same spacing as you find in DNA, 3.4 angstroms. And furthermore, you could add bases to the side because they're very reactive side chains, and those bases would also line up on top of each other because they have this kind of amphiphilic character. And then you could basically form a chemical backbone. These are all chemical steps that are highly plausible, not demonstrated experimentally though. And then if you had a change in pH, that backbone could break off and start folding up on itself, form a molecule that looks very much like RNA. That these chemical steps are all plausible, and furthermore, the building blocks are much more robust and likely in a prebiotic environment. And then eventually, RNA would take over, perhaps in a more protected environment, because it's more efficient than this particular mechanism. That's his idea. Untested. No one knows whether it's true, but it's just the kind of clever, creative idea. And the origins community is really open to people proposing ideas of this sort. Okay, step 3, conclusions. We haven't yet made a self-replicating cycle, but there are a lot of really interesting ideas out there, and I think it's going to be done fairly soon. I think this is something we can solve, but it remains a big mystery. And then, finally, step 4. As soon as you make a self-replicating set of molecules, natural selection takes over. Because you're making copies of yourself, you're competing for resources, there are slight variations in the organic chemistry of one cycle and another cycle, and so the mutations lead to competition and selection. And you may say, well, it's great for me to stand up here and say that. How can you possibly demonstrate that? I just want to briefly take you to Jack Szostak's lab at Harvard University, where he has shown how this can be done in a very elegant set of experiments, which are now used in industry to great effect in targeting viruses. What he does is he demonstrates molecular evolution by designing aptamers. Now, imagine this. You have an RNA strand that's 100 nucleotides, 100 letters long. That RNA strand will fold up in some complex way. you generate a random population of 10 to the 16th different 100 mers, strands, each one a different sequence of those 4 letters. And then you basically say, we want to give those molecules a task. They need to attach onto, they need to bind to another molecule, a molecule of a particular size and shape. And you attach that molecule to the side of a test tube. You dump a solution with that 10 to the 16th molecules into the test tube. You shake it around for a while. Some of the molecules stick. The RNA molecules stick because they have the right shape. Most of them don't. You dump out the solution. You wash it a bunch of times. The only thing that's left in your test tube are a few RNA strands that have stuck because they have just the right shape. You take those molecules and you go through a sequence of standard techniques in molecular biology to replicate and mutate them. And now you have a new set of 10 to the 16th molecules. In this case, every one a descendant of one of those molecules that stuck the molecule. And as a result, some of those descendants do it better. And you do it again, and you do it again, and you do it again, cycling around maybe 6 times, 8 times, 10 times. And when you do this, by the time you finish this process of just a few cycles, you have created molecules, aptamers, that are perfectly engineered, perfectly designed to lock on and target a specific molecule. In some cases, the same molecule occurs over and over and over again in different experiments. Out of the 10 to this, what is it? You've got 4 to the 100th power different possibilities of strands, and yet you zero in, you select the same molecule. The implications of this, of course, are profound for the case of evolution, because evolution, you see, is not random. Someone ever says, well, how can a random process lead to evolution? It's not random, it's selection. It's the opposite of random. Each selection cycle adds information to the system. And so here we have a process by which we can engineer molecules, molecular selection. Well, this mimics what happened in the geochemical environment of the early Earth. Cycles of wetting and drying, freezing and thawing, day and night, high tide, low tide. You had cycles occurring over and over again, molecules that were exposed to other molecules. Some would stick, some wouldn't. A selection process went on. Evolution went on. And so molecular natural selection has been demonstrated in the laboratory in this way. So, life's origins, 4 steps. Making the molecules is easy. Biomolecular selection, we don't know all the stories, but cell selection or mineral surfaces provide an answer. Big mystery here, the next step, making that self-replicating system, but I think we're close. And once you do that molecular natural selection, we understand how it happens. Okay, my conclusions. We don't know every detail. But there's no reason, I think, to resort to a supernatural description of the origin of life when we have so much chemical evidence that you can go through emergence from simplicity to complexity. This was a natural process. And that's the argument that we maintain, and we're going to continue working in this vein. I understand why this scenario troubles some people greatly. Again, how can we be created in God's image if we're the process just of chemical steps, of physical processes? And of course, evolution after that first step, the random selection, and who knows where it lived. That's troubling to some people. Yet I'd like you to think about the origin of life and the evolution of life in maybe a slightly different way. We live in a universe in which emergent complexity is all around us all the time. The universe started with the Big Bang. From the Big Bang came hydrogen, inevitably, inexorably. From hydrogen came stars. And stars, the processes of fusion and nucleosynthesis, and then explosions of new supernovas, seeded the universe with the entire periodic table of the elements. And from those elements came Earth-like planets. From Earth-like planets comes life. Perhaps inexorably from life comes self-awareness. A sequence of deterministic steps, not random, selected, And so we are living in a universe that is learning to know itself. Thank you very much for your attention.
Hazen, thank you for your thought-provoking talk. I think he has some idea of what minerals are when in 2008 there was a phosphate biomineral named Hazenite, in honoring our speaker. So if we could have some questions here. Dr. Mortimer?
I don't think that works.
Oh, does it work? I think so.
I think I'm moving loud enough.
Okay.
What strikes me as interesting in here is somehow underpinning all the processes that you've spoken about today, doesn't it lie in physical chemistry?
Could you repeat the question?
Yes, the question was, doesn't everything I've talked about today lie in basic physical chemistry? And absolutely. There's no single step here that isn't just a straightforward chemical experiment that you can do on a bench top, that you can make those chemical experiments by creating an atmosphere and subjecting energy to a mixture of simple molecules. You can take the molecules that are produced and look at various physical chemical properties of those molecules. You can, even what Jack Szostak does, every single step can be explained in an absolute classic way.
Why would you reckon that physical chemistry is not taught more basically in our curriculum?
Absolutely, and believe me, I take your point that if you want to study this or any other aspect of the biological and natural world, you absolutely have to have an understanding of basic physics and chemistry. And the processes we're talking about here are fundamental to every natural system. Again, deterministic properties. You cannot understand living systems, you can't understand geochemical systems without having the basic foundation, which is why I've been advocating in education for many, many years now, an integration of our undergraduate and graduate education. If you're in biology, you better take physics and chemistry and understand those principles. If you're in chemistry, you better understand biology. How can you separate these different fields from each other? So I take your point and appreciate your question.
I have a less sublime question, but what I'm wondering about with regard to the chiral selection of biomolecules, simple biomolecules, the crystal faces which bind to L-amino acids also bind to B sugars?
That's a wonderful question, and sadly, the answer is no. And indeed, crystal faces that bind to some L-amino acids do not bind to other L-amino acids. It turns out, and maybe this isn't a surprise in retrospect, this is my main research field, that it's highly idiosyncratic. Each mineral presents certain positive and negative charged regions on its surface with a topography. And whether or not a molecule sticks to it, especially chiral selection, which requires 3 points of interaction, is highly idiosyncratic if you happen to have just the right 3 positive and negatives on the mineral surface that match to the 3 negatives and positives on the molecule. In the case of aspartic acid on calcite 214 surfaces, it matches perfectly. You have a high degree of selectivity. Glutamic acid doesn't. And for those of you who don't know, glutamic acid and aspartic acid are sort of amino acid twins. You just have one extra CH2 group, which makes the one amino acid a little bit longer than the other one. So just the fact that the carboxyl groups, the CO groups, are a little farther apart means that it doesn't bond to the calcite surface. And we're, oh no, we want. I was so naive. I was sure I was going to find the magic mineral that you dump it into a solution of all kinds of amino acids and just the 20 biological ones come out. I thought that would be so neat. I mean, there's a science paper for you, right? No, it's not going to work. Yeah, so it didn't work.
You pointed out that intelligent design, even if you disagree with it, you said one of the positive things it did offer predictions could be falsified. I'm wondering if you agree or disagree that chemical evolution can't be falsified. For example,
Yeah, I take your point. I absolutely take your point. If we can show a positive way of doing it through very simple geochemical plausible processes, then we found the answer. But you're right. What if 500 years from now we've tried every possible conceivable combination of minerals and molecules and temperatures and pressures and pHs and all the other, well, I'm open to the possibility that maybe 500 years from now that'll be the result. But I see progress being made so rapidly and so many really exciting results coming out of different laboratories, new molecules being formed, new ways of selecting molecules, that I'm optimistic. But as a scientist, I have to agree with you that there is also the possibility that 500 years from now we will just throw up our hands and say, well, we've tried everything and we can't figure out how it works. And let's face it, as scientists, we just have to keep an open mind and keep trying. Obviously, I wouldn't spend my life working on it if I thought that was true. Now, if I can make one more point, you still have three possibilities for this origin of life. Life could have been a miracle or intelligent design. And it could have been a cosmic imperative, in which case we'll find the answer somehow. It's also conceivable that given the fact that our universe has 100 billion galaxies, each with 100 billion stars, and so there very easily could be 10 to the 20th Earth-like planets, that given sufficient time and sufficient number of planets, even if it's highly, highly improbable, but consistent with laws of chemistry and physics, you might get an origin of life. Or maybe on Earth, origin of life happened multiple times, but just once every 100 million years. You know, and so it's very rare. And if it's a very rare event, even though it's completely consistent, we'll never reproduce it in a laboratory simply because we don't have the time and the combinatoric richness of a planet over 100 million years. Do you want to take my point? So that's another option. So I'm afraid that philosophically, scientists will never concede that it had to be intelligent design simply because we recognize that time and combinatoric richness of environments is something we can't reproduce in a laboratory. So it's a very interesting philosophical question that you raised, and I'm glad you asked it.
500 years is not a long time. After brilliant physics was established, there were problems, very simple problems about that you mentioned that it couldn't solve for 200 years. But people didn't abandon Newtonian physics because they felt there was an explanation, and they did find it.
Yes.
And I think what you're dealing with is far more complicated than the problems that they were dealing with in Newtonian physics. So I could say 500 years.
Yeah. And I will also add that not only is it 500 years is not a long time, but there are very few of us doing this research. That the amount of funding for origins research compared to, say, biomedical research, which may have some peripheral relationship, it really doesn't focus in directly on the point, is very small. So if in the United States right now there may not be more than 150 or 200 people who really are working at a benchtop thinking about origin of life chemistry. And that's a small number of researchers given the combinatoric richness of what we're finding in the natural world.
A couple of comments. You were discussing Shostak's work, and you mentioned that the end product of the cycles of selection were perfectly fitted to their ligands. And that's not true. They're not perfectly fitted. There's a range of affinities.
Yes, that's true. There is a range.
Solutions to combine
Yes.
the same ligand. This is true both for
Yeah.
aptamers as well as for peptides and proteins that are selected from libraries to bind a particular ligand.
Right. Yeah, I understand what you're saying, but Carruthers' work on the GTP aptamers, what he found that there's certain classes of solutions that always come up that are highly fit, that have certain subsets that are highly conserved. So you're right. I mean, it's not.
Depends on the complexity of the ligand.
Yeah, in the case, I'm thinking specifically of that early work on GTP binding.
And in reference to this other question about the difference between intelligent design and kind of what you're doing, at each step of the way they're making testable predictions that can be falsified. And intelligent
Absolutely.
design basically has been falsified. Because the one kind of prediction it makes is that if you delete one component of what is referred to as an irreducibly complex system, that the system will lose all function. Now, they don't really have a precise definition of irreducibly complex, but the founder of that idea has said the immune system is irreducibly complex.
You're right.
And numerous genes have been knocked out, and it doesn't destroy all function.
Yes.
So it has been falsified.
And the subcomponents of a flagellum also have high degree of functionality in other contexts. So you're absolutely right about that. But I'm just trying to be as respectful as possible to what other people are saying. And I think as scientists, we need to sort of be really recognizing ways to legitimately falsify ideas that are out there. And it's a difficult debate because, of course, on the one hand, some scientists are very convinced that the scientific method is the only way to arrive at truth. I mean, we believe that science works. And on the other hand, there are people that believe that there are other more valid ways to approach understanding. And I like to find some way to form a useful debate and dialogue in that whole structure. Yes.
This past summer, we read as a common reading Daniel Quam's book on Darwin. And I came away with that one short sentence in there that might seem relevant now. I wish I could reproduce it exactly. But the essence of it was that Darwin's challenge in evolution was basically the supremacy of the human. That if he's correct, there is, what's the difference between a worm and a rabbit, and or are we human beings just another animated form of meat that come on this planet for a while?
You know, I can understand why that's a question that disturbs a lot of people. And on the one hand, science can just say, oh, we're just another step in an evolutionary process and it's just a continuum. However, I think we can also come up with a maybe slightly different answer, which is more satisfying in terms of those deep questions about meaning and purpose. I'm not saying that science can answer those questions directly, but I've talked to you about emergent complexity. And if rather than thinking that the centricity we're looking for is a physical centricity, the center of the universe, or it's some kind of physical or temporal center, but rather we think of emergent complexity as those systems which we find most interesting, that we find most diverse, perhaps most complex in terms of local organization, and we say we value those systems because of their intrinsic behaviors, then by far the most complex system, the one that has achieved the highest level of emergence that we are aware of, is right here. And I think that's something, while it's difficult to quantify right now because we don't have a really good definition of complexity or functional information is an expression that we've used. But nevertheless, and by any subjective measure that you have, the most concentrated information per volume in the universe that we know about is right here. Not in a worm, not in a single cell, it's in the conscious brain. And so perhaps that in some way maps onto some people's perception of why humans are special or they're different. That's a quantifiable aspect of us that is, in that sense, superlative. Now, maybe to a lot of scientists that's not important, but to a lot of people who want to feel that somehow we are special, that we are separate, that is a quantifiable aspect that's separate.
Well, thank you, Dr. Hazen. If you have any other questions, you could come afterward and speak with him. Thank you again.